Good afternoon, everybody, and welcome to today's RSSL webinar hosted by Tim Sandle and myself, Lyndsay Cooke. So as highlighted on the screen, today's webinar will be focusing on mycoplasma contamination in cell cultures and ATMPs. Before we begin, please can I go through a few housekeeping points with you. If you have any technical issues, please send a short message in the question box on the right-hand side of the screen. Similarly, during the seminar, if you have any questions, please do post those in the question box, and we will try and cover as many of the questions at the end of the seminar, time permitting. If we are short on time, we will take your questions away and reply in the days following this event. We will also share the slide deck from today with you all via email. So as a very quick introduction to myself, my name is Lyndsay Cooke and I'm Laboratory Manager for the Biomolecular Analysis team here at RSSL. I've been with the company nearly 5 years and I'm a viral immunologist by background, having worked on infectious viral diseases of livestock for numerous years. Please feel free to reach out to me about any of the analytical testing questions that you may have or any of the services that we will describe today, and I will happily support you through your testing requirements. Before we begin with today's seminar, I will very briefly highlight RSSL's capabilities and the brilliant work that we do. So first and foremost, who are we and why are we different to other CROs? Well, Reading Scientific Services Limited, RSSL, are a contract research organisation who strives to provide high quality and innovative analytical testing services to the food and pharmaceutical sectors. We are an independently run CRO, but are parent owned by the global snacking brand Mondelez, who you may recognise as the owners of Cadbury and Oreo, to name a few brands. This gives us support and financial backing as our markets grow and of course continue to diverge. We are proud to share that we have won many awards, including for the services that we offer as a CRO, but also the culture of support that we have in developing our staff. We have been running as a company for around 30 years and are based in the Reading and Wokingham area, which for those of you that don't know is approximately 40 minutes west of London. On the pharmaceutical side, we have a global client base ranging from big biopharma companies to small biotechs too. We have 4 sites hosting approximately 250 highly skilled scientists covering multiple areas of expertise. And this enables us as a company to provide a diverse range of complex testing, all within our extensive interconnected laboratories. And to highlight further on our laboratory space, we were lucky at the end of last year to move into a brand new 26,000 square foot building on the University of Reading campus. And this has enabled the company to grow and expand the scale and breadth of our service offering. I should note as well, our laboratories are MHRA and FDA approved, enabling us to provide GMP accredited testing, including method development and validation. So now further focusing on our biopharmaceutical services, this slide is very busy, which I will not go through in depth. However, hopefully it highlights the breadth of testing that we can offer, ranging from biomolecular, microbiological, and immunological analysis. This offering enables us to test traditional protein-based biopharmaceuticals, but also new and novel therapies, including lipid nanoparticles and cell and gene therapies. We can support at all stages of the drug life cycle, from drug discovery or early phase development, right through to commercialization and finished product GMP batch release testing. To keep up with client expectations and regulatory guidelines, we are continuously innovating and expanding our service offering. For example, we have recently launched mass spectrometry testing for the detection of host cell proteins, bringing our service offering in line with the imminent USP recommendations and to complement our existing ELISA service offering. So if you can't see a service listed here, then please do reach out to me and I will be happy to help. Given the nature of this seminar today, I'd just like to draw your attention to our biosafety service offering, which is on the right-hand side of the screen. And this service offering plays a critical role in ensuring the microbiological safety of our clients' products throughout drug development, manufacturing, storage, and ultimately the end use of the product. Tim will touch upon mycoplasma detection methods in today's seminar, and I'm pleased to share that RSSL are currently in the final stages of launching its in-house qPCR-based mycoplasma testing service. So please do get in touch if you have a mycoplasma testing requirement and would like to be added to our waiting list. Furthermore, we are well placed to support testing in line with Annex 1 regulations and have a highly knowledgeable technical team who would be more than happy to help and advise. Additionally, we're also in the process of launching rapid sterility testing, which is something to watch out for in 2025. If you would like any further information on the testing services discussed, then please do use the link at the bottom of this slide. Equally, as highlighted, please do reach out to me and I will be happy to help and highlight how RSSL can help you. So please can I now introduce Dr. Tim Sandle. So Tim has got 25 years experience in the field of pharmaceuticals and is a member of several editorial boards and has an extensive publication record. Tim also works in the pharmaceutical industry, so we're looking forward to some insider knowledge today. So thank you very much, Tim, and I will pass over to you.
Okay, thank you very much, Lyndsay. Okay, I'll just switch the display. There we go. Okay, right. Well, thank you, everybody, and thank you for attending today. So what we're going to be having a look at are a brief introduction to mycoplasmas. We'll be considering why mycoplasmas are a concern for cell therapy products. We'll consider some of the specific contamination risks and be considering how these risks arise. We'll be considering some preventative measures, some techniques for contamination removal. We'll have a look at test methods and also consider testing strategies. So just to begin with,
sorry, just to interrupt. I can see your GoToWebinar dashboard, just so you know.
Oh, okay. 1 second. Let me try and move that.
There we go, it's gone. Thank you.
Okay, thank you. Okay, so we're considering what are mycoplasmas. So mycoplasmas are bacteria. They're a specific genus of bacteria, and they're defined by a specific morphological characteristic. And this is the absence of a rigid cell wall around the cell membranes. And this is the derivation of the family to which they belong to, which is the mollicules. And this derives from the Latin word mollis, which means soft or pliable, and cutis, meaning originating from skin. And mycoplasmas are distinct from other bacteria that might also go through phases of not having a rigid cell wall. So mycoplasmas permanently don't have a rigid cell wall, but there are certain bacteria called L-form bacteria where under certain conditions, specific strains of bacteria, which might under one set of conditions have a cell wall, under another set of conditions have the absence of a cell wall. And the mycoplasmas have their specific characteristic because they are missing a core element that's found in most other bacteria, which is peptidoglycan. That's the cell wall material that makes those bacteria that stain gram-positive, and this is missing from the mycoplasma, and instead they have sterols in their membrane that give them a basis of some structural characteristics. Okay, so further with mycoplasmas, they are also regarded as the smallest free-living organisms. They are also classed as parasitic because they enter target host cells and then use the cell machinery in order to either multiply or to enter into some form of hibernation until conditions are suitable. Their flexible cell membrane gives them various sort of shape-shifting properties, and this is one of the properties that becomes problematic when we consider filtration as a removal step for mycoplasmas. They also have a very small genome, so the typical mycoplasma genome is around 0.6 to 0.2 compared with a bacterium like E. coli, which would be 4.6. In terms of their generation time, it's between 1 to 9 hours, but before the growth of mycoplasmas kicks in, they have quite long lag phases. So it's for these reasons, if we're trying to culture mycoplasmas on culture media, we need at least 1 week of incubation in order for colonies to be visible. And because of their small size and relative sort of plasticity, they will pass through a 0.45 filter, and in certain circumstances they will pass through what is traditionally regarded as the sterilizing grade filter, which is a 0.22 rated filter. And they're also resistant to quite a lot of antibiotics, and this is because most antibiotics target the bacterial cell wall, and then obviously the absence of a cell wall means that quite a lot of antibiotics are not effective, and this precludes the use of beta-lactam antibiotics in particular. It's also possible, as with other bacteria, for mycoplasmas to form biofilms as they attach to solid surfaces. So, and that's particularly a predominant characteristic on glass and plastic surfaces. And as we have discussed in previous talks, once microorganisms attach to a surface and start to form biofilm communities, it confers an additional level of resistance and gives them a degree of resistance to environmental stress and to disinfectants. And in terms of human pathogens, some species of mycoplasma are pathogenic to people, so they can selectively infect the lungs and other parts of the respiratory system. They can cause skin infections and urinary tract infections as well. So of the couple of hundred species of known mycoplasmas and related organisms, around 18 are considered to be potential pathogens of humans. So Mycoplasma pneumoniae, Mycoplasma genitalium, Mycoplasma hominis, Uriplasma uriculatum are some examples of those pathogenic species. Probably the most widespread within the community is Mycoplasma pneumoniae, particularly as a cause of some respiratory tract infections, particularly in children. So in terms of certain biotechnological applications, mycoplasmas are undesirable, but they're also can be relatively common contaminants of both human and animal cell lines, and often the detection of them can prove challenging. And the reason they're undesirable is, other than just being a general adulterant and impurity, is because they can enter the cells of our cell line, and they can then alter the cells they've infected at the molecular level, and this can trigger changes to cell morphology and alterations to growth characteristics, and also in any generation of substrates that we wish our cells to produce. So they can ruin an entire cell line, and cell lines are of obviously great importance to quite a lot of current biopharmaceutical manufacturing and research, particularly in the area of producing advanced therapy medicinal products. So the kind of concern extends, as we've discussed, partly because contamination of cell culture can't be visualized. So mycoplasma contamination does not produce visible turbidity, as we might see with most other species of bacteria or fungi. As they grow under appropriate nutrient conditions, you can often see turbid growth after a period of time. We don't have that option. And it can be, if we leave the detection too late, not only do we have the risk of infected cell line, but we could then start processing that cell line and then not produce the end product that we are keen to produce. And this is a particular problem with the emerging human cell-based medicinal products. And these are, obviously represent a lot of cutting edge medicinal development. So we may have cells that are self-renewing stem cells. They might be more committed progenitor cells, or they might be terminally differentiated cells that we're seeking to get specific physiological functions. We might be using cells originating from the same patient, or we might be using allogenic cells from a different patient. We might subject these cells to some form of genetic modification. So there's a variety of different types, and they're all prone to potential mycoplasma contamination. And that's what we need to focus on and avoid. But we also have some other challenges, particularly with the ATMP products. These are often very expensive products, so the ruination of a batch carries very high consequences. But we're also often producing batches in quite small quantities, particularly where patient specific, or we're using autologous cells, we're generating very small quantities of final product. And often our products will be having very, very short shelf lives, and sometimes even the window between the final harvest and transfer to a patient can be as short as a matter of hours. So the consequences of infection with mycoplasma is incredibly high. So in terms of sort of general quality control checks, it's important that the cells used in the process have been assessed for viability and also the general integrity of the cellular functions. The detection of possible changes at the cellular nature could influence the intended function. So what I was saying earlier about if mycoplasmas infect the cells and cause those changes, then that can have catastrophic implications for the final application. And we have different ways to assess viability. So we can use viability dyes, we can use manual or automated analysis, we can use flow cytometry, and so on. But our primary goal is sort of minimizing contamination in relation to the cell bank. And we want to have a degree of control as the first step to minimizing contamination by ensuring that no other living or infectious materials such as viruses, other cell lines or cell strains are going to be handled simultaneously in the same area or not without a degree of protection or some degree of control. So mycoplasmas, if they do enter cell cultures, can spread quite vastly. With cell cultures, most mycoplasmas present a risk by remaining attached to the surface of cells or invading the cell. And we've seen this through various sort of ultrastructural studies, which highlights these kind of twin risks with mycoplasmas. And the mycoplasmas will want to connect to the cells in whatever cell line. And this goes back to what I was saying about their parasitic nature, because often mycoplasmas are unable to perform the same functions that other bacteria can perform. So they're invariably dependent upon the host cells for their nutrients. So although the biggest risk is with mycoplasmas changing the shape, the cell morphology, I've also mentioned about the risk of them altering the cellular metabolism, so we don't end up with the end result that we're seeking. But there are also some species that are cytopathic, which obviously means they kill the cells as well. So each one of these risk mechanisms can lead to the ruination of the entire cell line. And the kind of major problem presented by mycoplasmas is with their ability to adhere to cells, and it's this adherence that is the sort of major virulence factor. And mycoplasmas have developed various genetic systems enabling their attachment, and they also tend to have a fairly varied set of surface proteins that enables them to attach to a range of different cell types. So they have what's regarded as a strong versatility of their cell coat, as well as size variation. And they, in the mammalian body, they're also fairly adept at immune system avoidance. So in terms of considering sources of contamination, these are going to vary due to the fairly ubiquitous nature of mycoplasmas in the environment. So we can have people, materials, consumables, equipment, culture media and reagents we add, as well as the inherent cell culture itself. Droplet generation during handling of cell cultures is a relatively common mechanism, particularly from human operators. And another risk factor in the general environment is, although mycoplasmas often need to hijack cell material in order to grow, they're also fairly hardy in their ability to survive for prolonged periods under dry conditions. So if we're not practicing good disinfection, or in relation to the risk of droplets and controlling sources that generate aerosols, if we're not practicing good aseptic techniques, then these can be quite common mechanisms for infecting cells. So because of the difficulty in detection, the impossibility of visual detection and the range of different sources, then it's incumbent upon us to use reliable mycoplasma detection methods and to test cell cultures on a regular basis in order to limit the impact that any contaminants might have on subsequent processing. And we'll have a look at some of those shortly. And it's also good practice to keep incoming cultures in quarantine until we've undertaken some initial tests to show that these are mycoplasma free, and to immediately discard any contaminated cultures. And we can gain greater assurance of this if we're purchasing cultures from reputable suppliers. And one way of trying to control this is potentially through the use of antibiotics, but this can also create problems because it can promote antibiotic resistance, and it can become counterproductive. And then further with personnel, we touched upon aseptic techniques, but wearing a mask and thoroughly washing or disinfecting gloved hands is an important control mechanism. And then once we're actually into processing, then we need to have good control of our manufacturing steps in order to prevent contamination. And the same principles of whether we're dealing with viruses, bacteria, fungi, prions, and so on, it also applies to mycoplasmas. So we need to have that control, and we also will, at periodic intervals, depending on the complexity and length of time of our process, to be able to employ techniques that can minimize mycoplasma contamination occurring in the manufacturing process. And one key feature is the processing environment. So the measures that we take to protect the air through an effective heating, ventilation and air conditioning system, and also the design features in terms of either using single-use disposable technology or the way that we process reused equipment, and in avoiding cross-contamination, particularly with equipment that we've already used for one batch not interacting with another batch. And although we may have filtration as part of our process, particularly if we're trying to produce a sterile product, we do need to be mindful that mycoplasmas, based on their cell size and flexibility, can penetrate sterilizing grade filters. So these are the filters of 0.22 micron, which I mentioned earlier, and it's to do with this plasticity that they're capable of sometimes penetrating these filters. So if we wish to use filtration for mycoplasma clearance, then we need to be considering 0.1 micron rated filters. Of course, the downside of that is that we then could end up with greater yield losses. It's also important, if we're purchasing filters of that specification, that we have evidence that the filter manufacturer has used mycoplasma cells under representative conditions of use. Another thing that we need to be mindful of, particularly if we are engaged in sterile manufacturing and we're making an aseptically produced medicine and we need to execute media fills, which is a common aspect for aseptic manufacturing activities, is a lot of culture media used for media fills, particularly tryptophan soya broth, is often contaminated with mycoplasmas. So again, ensuring suitable quality control of that is important, because the complete removal of media residues in itself can be challenging. So where cell cultures have been contaminated, then other attempts outside of filtration can be used to try and remove mycoplasmas, but this is challenging and complex because we're obviously seeking not to harm or damage the human or animal cells. So we have filtration, as we've discussed. We also have the potential for ultraviolet light, and we have the ability to use selective antibiotics if we feel that is suitable, though the widespread use of antibiotics, as I said before, can be counterproductive. And where continuation cannot be addressed, then a reliable, we need to make sure that if we believe we've got an infected batch or an infective starting culture cell line, we need to dispose of that. We should undertake a reliable decontamination step before we dispose of the culture. Otherwise, we do have the risk of either putting contamination into the environment or having some kind of event that leads back to cross-contamination. So decontamination by heat is generally the preferred method. Mycoplasmas have a low level of heat resistance, so the typical, say, autoclave cycle of 121 degrees Celsius, 15 minutes is normally effective for this purpose. Okay, so we move on to detection methods. And for many, many years, the standard method was culture using a supplemented agar or a broth medium and attempting to recover any mycoplasmas present under appropriate atmospheric conditions. And this was relatively long-winded. Spoke earlier about the sort of minimum 1 week incubation because of the long growth times required. And also there's limitations as with the trying to recover any bacterial species, in that some are simply uncultivable under the standard conditions, yet they could be contaminants of cell lines. So a typical culture medium to stand a chance of growing mycoplasmas would be one that would normally consist of heart infusion, peptone, yeast extracts, salt, glucose, maybe arginine, and the addition of horse or calf serum. And often an antibiotic would be added, one that was not active against mycoplasmas but would inhibit the growth of other bacteria, because one of the challenges also the cultural method is if other bacteria are present, then the chances are these will grow faster, and the plate could be covered with a staphylococcus before any attempt could be made to try and count the mycoplasmas. Fungal growth as well is another risk with the cultural technique. And also there's some variability with some of the culture media because of the complexity of the medium. This can also lead to some variability in its manufacture. But where the cultural method is used, then a typical approach is an attempt to grow mycoplasmas in broth and then to transfer a portion of the broth into agar and then incubate those plates. Colony formation is microscopic, so typical colony sizes are within the sort of 100 to 400 microns. So an attempt needs to be made to count those colonies or visualize them using a microscope, and we'll be looking for this kind of characteristic fried egg type appearance. Another challenge is that colonies are often embedded beneath the surface of the agar. Now we need to get the optimal cultural conditions, so we'll be trying to create a microaerophilic environment. So that's nitrogen with 5 to 10% carbon dioxide at an optimal incubation temperature, so one that's closer to the human body temperature of 35 to 38 degrees Celsius. And to make absolutely sure, a 14-day incubation would be advisable. So we may start to get contamination detectable at 7 days, but because of some fastidious slow-growing organisms, then 14 days would be optimal. So there's limitations with cultural methods, and in contrast, faster diagnosis can be obtained through techniques like PCR, which we'll have a look at in a minute. The other thing that's important, and again it applies to any form of bacteriology, is to ensure that the culture medium is suitable, certainly for each batch before use. So we would need to use positive controls, and we would need to select a suitable range of mycoplasma from different sources. So that there's some examples on the slide of the types of organisms, and this is taken from a European Medicines Agency document that are good screeners of mycoplasma culture media. And these species are antibiotic sensitive, they're fastidious, they have different growth rates, they're likely to be contaminants, and some of them possess pathogenicity against the types of cells that we're seeking to screen. And again, with any screening of culture media with bacteria for any other test described, then we're seeking a low level challenge, and the pharmacopoeia definition of low level challenge is less than 100 colony forming units. We also should undertake method validation to prove that we can recover mycoplasmas in the presence of the product on the cell line. And if the validation is not successful, so we can't recover the challenge population, then we should seek to neutralize any inhibitory substances or perform dilutions, assessing that obviously we're not going to dilute too far that recovery becomes impossible. And if we are going to use a neutralizer, then we need to be sure that the neutralizer itself is not toxic to mycoplasmas and then what we recover is accurate. We also have staining, which can enhance the culture method and lead to more accurate counting. And the common staining method is a DNA fluorochrome method, a bit such as Hearst stain. And this uses a blue fluorescent dye, and it's designed specifically for staining the nuclei of living or fixed cells. And this stain can be excited by ultraviolet light of around 350 nanometers, and visualization is then attempted through fluorescence microscopy, and samples infected with mycoplasma will appear as fluorescent nuclei, as will any extra nuclear mycoplasma DNA. The limitation is that we really need to be trying to see around 100 mycoplasma cells or more in order to enable sufficient staining. So it's something that's quite powerful for validation. It can be quite powerful where we have higher levels of contamination, but it is weaker when we have lower levels of mycoplasmas that we're trying to detect. Dark fields microscopy can also assist, but there are still limitations. So in recent years, pharmacopoeia and regulations have shifted to allow for alternative methods, and the main regulations for screening for mycoplasmas in US and in Europe are shown on the slide there, and I won't go through those now, but you can we can use those as references to read up on. So the big advancement was with the advent of PCR, and it was really during the sort of mid to late 1990s that we started to get PCR assays for mycoplasma screening. And prior to PCR, then there were also some innovations in species-specific antibodies using either immunostaining or ELISA assays. But the PCR test took things forward quite considerably, and the PCR testing is based on the detection of 16S, sometimes 23S RNA molecules, which are common to all species of mycoplasmas, and a commercial test kit would allow us to extract this genetic material for testing, and then the PCR method would then obviously allow the acceleration of that genetic material and produce sufficient quantities to allow us to carry out scintillation counts.
I think.
Okay.
Yeah, from that one.
Okay, so just very briefly on this slide, it's just showing the area around the chromosome that we're trying to target with the PCR. So it's where this highly conserved region is, which is the 16S RNA region. And then on this slide, I was talking about the validation of the PCR. So running a validation study is important. It's important to directly challenge samples with mycoplasmas using type cultures drawn from a reputable culture collection, such as the American type culture collection, and to run tests in triplicate. And at various time points to undertake the necessary DNA extraction and PCR amplification. And it's also important where we're undertaking dilutions to also evaluate those, and to introduce a level of robustness, as well as running things 3 times, is to have 2 or 3 different analysts repeat the activity. An additional challenge is that some methods don't allow accurate discrimination between viable and non-viable mycoplasma contaminants, and then this can lead to false positive results. So if we use a raw material that's got inactivated dead mycoplasmas in there, there's a risk that certain methods will pick up mycoplasmas that are not actually viable and therefore not of necessarily any harm. So again, understanding our ability to detect that is really important. It also stands that the sensitivity of PCR can sometimes be enhanced through a pre-enrichment test. This is also something that's important with the culture method as well. And this again just sort of helps get sufficient cell density, so we stand a greater chance of detecting the contamination that we're seeking to detect. And although we have PCR, it still remains that the pharmacopoeia contains the culture method, and there's reference to the culture method still being a test for adjudication. What also the European pharmacopoeia is saying is that it allows alternative methods to be used as well. So we have wider options. And some laboratories elect to run both methods, and others have performed all the necessary validation in order to switch to PCR. And where we're considering alternative methods, one important consideration is the GC to CFU ratio. And this is something that can be used to distinguish free-floating genomic DNA from that of live mycoplasma. And the GC to CF ratio for each culture, each individual mycoplasma is going to vary due to the growth rate and culture condition variability. So we do need to have some understanding of what we're using, particularly for validation. So ideally, we want to use mycoplasmas that have low GC to CFU ratios, because this is kind of the most robust method for detecting contamination. And this can also help with the differentiation between the sort of cultivable and non-culturable mycoplasmas. So a lot of thought needs to go into the detection and the types of strains that we're going to be using for our positive controls, that they are truly representative. They have this low GC to CFU ratio, as I mentioned, and that we're sure that viability is maintained as we challenge our samples. An example of sort of the alternative methods to PCR and cultural methods are microelectrical mechanical systems, or MEMS. And these are microarrays DNA chips which have evolved from southern blot technology, which can also help with mycoplasma detection. So that's test methods. Just quickly to finish up with, another important consideration is a thorough testing strategy. So I mentioned earlier about the risk to the master cell line. We've also got any subcell lines, and we've also got all the different manufacturing stages. So we need to understand when we're going to test, why we're going to test, and how we're going to test, and to and make sure that we've undertaken appropriate risk assessment and looked for potential weaknesses in the system. So representative test points are important, but also representative samples as well. So given that mycoplasmas are contaminants that might adhere or be present in cells, so samples should contain both cells and supernatant, and or we need to qualify that we're just going to look at culture supernatants alone. But we've got to make sure that the samples we are testing are suitably representative, they're either homogeneous or we've undertaken all the necessary steps to try and recover any mycoplasmas that might be present. Okay, so that brings the main part of the presentation to an end, and we can move on to Q&A.
Lovely. Thank you very, very much, Tim, for that insightful presentation today. We have had a few questions come through, so I'll kick off if that's okay. So you described cell culture risks in depth. One question that has been highlighted is throughout the cellular banking process, how well do mycoplasmas survive through freeze thaw cycles?
Yes, that's a good question. In most cases, they won't survive. I mean, some can survive, but it depends on the way the freeze thaw is designed. So generally, the sort of a steady progression of freezing is most effective. Sometimes with a very rapid freeze that can actually protect mycoplasmas, whereas a steady rate of freezing is actually more effective at sort of killing cells. So in general, effective, but there are certain circumstances, and it would also depend upon the medium as well, in terms of what sort of some media can act as a kind of cryopreservative. So
Okay.
again, it's something that sort of test data can provide answers to.
Okay, lovely, thank you. And so sticking with the theme of cell culture, then you described serums as a possible mycoplasma risk or source of contamination risk. How common are mycoplasmas within serum, and if a source was to be used that wasn't screened for mycoplasma, would there be a high risk of infection there?
So I think there's been obviously steady improvements since the 1990s. So I think the rates of contamination are not as high, but and I think that the answer depends a little bit on whether we're talking about established cell lines or sort of more novel cell lines, and the where these are obtained from. So it's kind of a tricky question, but sort of literature surveys are pointing to sort of still around sort of 20 to 25% of cell lines are potentially contaminated. If there's not screening undertaken and then there are mycoplasma present, then again it depends upon what the application is, but there's a real chance that that infection will be quite damaging and cause unwanted physiological changes to the cells.
It's still quite a high statistic, isn't it? 25%. So yeah, very interesting. So what factors would you suggest for consideration then when sterilising serum, if that was something that that the audience wanted to do in order to try and remove the mycoplasma present within serum?
Yeah, well, probably the only really effective way of sterilising is through filtration, ideally at sort of 0.1 micron, because trying anything else does carry the risk of, you know, anything that's going to kill mycoplasma will probably damage cells. But you can do some applications of UV light. You've got to be very careful and know, you know, the point that you might inactivate mycoplasma, but you won't harm any other cells in the line. The only other recourse is to target an antibiotic, but that really depends on sort of only having one type of mycoplasma contaminant. If you've got multi ones, then it's unlikely you'll find the right antibiotic. And then I think the other point I was making was that you know, if we have too much of an indiscriminate use of antibiotics, then we're eventually going to create a condition for ourselves that we don't want, which is, you know, wider spread antibiotic resistance.
Lovely, thank you. So one final question then, Tim. I know we're at three o'clock now, but the final question being, are personnel the greatest risk factor for mycoplasma contamination?
I think they're the greatest risk factor if we're buying in cell lines that we know are pure and free of mycoplasma. Then often it's people would be quite a significant route of cross-contamination, either through general practices like not disinfecting correctly, through handling, or through you know, droplets by sneezing or coughing inappropriately. So good aseptic techniques are always really important.
Brilliant. Thank you very, very much, Tim. So as mentioned at the start, all of the questions that that were posted will be answered as a follow-up from today's session. Finally, I just want to leave you once again with my contact details, and also just to highlight at the top there the upcoming conferences that myself and my RSSL colleagues are attending in November. If you are attending any of these events, then please do get in touch, and we would gladly arrange to meet with you and discuss any testing requirements that you may have. So lastly, that just leaves me to thank the audience, and thank you of course, Tim, for the brilliant session today. Thank you very much.
Thank you.